CA plots by changing some of the parameters in the autoplot function. How did that go? Did anybody run into any errors or notice anything interesting? Did anybody set alpha to 0 and see what happens there? Yeah, Evelyn says yes. Yes to which part and what happened? Alpha equals 0. All right, so alpha changes the saturation of the label dot colors. Yes, that's right, Eowyn. So Evelyn, when you set alpha to 0, what happened? Disappeared. Yeah, exactly. So alpha equals 0 means no color at all, whereas alpha equals 1, alpha equals 1 means all possible saturation, all possible color. So, okay, perfect timing because I have to rerun all my cells. Hi, Sylvan. Welcome. Now it's time to talk to the students.
Okay, guys. I'll put my video on for a few minutes, but it's going to be a bit hectic here because it's lunchtime, so I'm sorry. There's going to be activity coming up very soon. Yeah, I wanted to just drop by and say hi to everyone. I'm the project manager for GeneLab, and I just wanted to make sure you guys were having a great experience. And also, I'm really excited about this program. It's our first try. So, we're looking forward to your feedback as well. Thank you for, joining us. I don't know if you want to throw me questions or things, or, I'm curious to see are you guys happy with the program? Is there like a thumbs up, thumbs down on this platform that they can use? Cool. They read the thumbs. Oh, I like that. I see Phil is clapping. Good. Oh, wow. That's great. You're at the end of the training, so I'm taking this as that you feel like you've learned quite a bit. I'll wait for the reactions, see what happens. Maybe they didn't learn that much. Oh, yeah, someone else.
Can you see the chat, Sylvan?
Yeah. Okay, yeah. So informative. Def, learn a lot. Yep, learn a lot. That's great. So, the rationale behind this program was really kind of to kill two birds with one stone, being able to train people on bioinformatics, which is super exciting, but also giving you the opportunity to really understand the some of the nuances and uniqueness of a data set that comes from space sample. And you can see how we have very unique challenges. I think the biggest challenge you probably notice is the lack of strong statistics, a small number of specimens being flown on the space station. Those are, issues. And I'm really looking forward for the next generation of scientists coming along, because I feel like there's so much to still do in terms of statistics and new method, new approach, better experimental design to try to address this question. So I hope you got a glimpse of this, and it's a seed that we're planting that I hope will lead to some exciting things for some of you. I don't know, based on this bootcamp, do you feel like you want to spend more time researching more on space biology? Yeah, Evelyn Wong says yes. Or yeah, I see that. Cool. I love this stuff. Excellent. Veronica, Felix, cool. Well, if you have any question, let me know. Otherwise, I will jump out. I'm sorry it was very brief, but I don't want to take on your bootcamp time.
If anyone has any questions for Sylvain, feel free to put in the chat or unmute. There's a question in the chat.
Yes, I agree. Bigger rockets would be great, right? It's actually not the rockets, it's the space station that's kind of like they kind of crammed in there. So ideally, it would be nice to see that. Actually, it's funny. I have the same fantasies to create some kind of like a gigantic CubeSat that would be self-autonomous, where you could actually put the mice in there and they would survive. There were missions like this in the past. Bion is one of them, a satellite mission where they leave the animals in there alone. The bad news is when that mission, I think it was a Russian mission, when the animals came back on Earth, half of them were dead, so it didn't work very well. So some progress to be done here. I'm curious about the physiological impact of space on the body, and this has been an eye-opening experience. Cool. Yeah, I think that's one of our goals too. One of the big questions is how relevant is what we do with mice to humans. And I would not say only mice. Drosophila is another very good model. You probably learned that you can use the Drosophila to look at the immune response, or even the central nervous system, so behavioral, neurological, cognitive functions are also studied with Drosophila. So there's quite some research interest for us in terms of scaling things from animals to human. Cool. Well, it looks like I answered most of your questions. Again, thank you for joining us, and thank you, San Jose State University, for going along with this vision. I'm super happy that it went well, and have fun. And I hope you have a great end of your session. Hey, Phil.
Thanks for coming by, Sylvain.
Of course. You take care, guys.
Thank you.
Thanks, Sylvain. Bye.
Thanks, Amanda. Thanks, Lauren. Yeah, you can clap for those two. They work their tailbone off for this, so, and Phil too. So, all right, see you guys.
All right.
Good job, guys, with the compliments. Exactly what we wanted.
Yeah, you just made us look really good to our boss. Thank you so much.
I did want to just mention another thing about expanding the space station that Sylvain was talking about. So that it's not the rocket, it's actually the space station that's limited in space. And there is a company, Bigelow Aerospace, that's working on inflatable space habitats. So their goal is to launch these habitats that once they get to the space station, they can be inflated from within the space station outward. And kind of the big long-term goal of it is to be able to have a whole animal facility in one of these kind of inflatable modules attached to the space station. So ideas in progress for things like that.
That's really cool. I didn't know about that. All right. Let me just share my screen here. Oh, I am sharing my screen. Good for me. Okay. All right. So we were talking about Autoplot. I just wanted to share something, you guys. We learned about how we could rerun all of the cells in a Jupyter notebook by clicking on a cell and then going to kernel, restart kernel, and run up to selected cell. I just wanted to point out, in my experience, as of 5 minutes ago, this won't work if one of your cells is designed to produce an error or produce an error by accident. So we had you run a cell up here that produces an error. And so when I was saying, oh, rerun everything after this cell, or through this cell down to the cell I had selected, it was erroring out here and then stopping and not continuing past the error. So this works really well when you have a nice, clean Jupyter notebook that runs without any errors, which is usually what's going on. But in this case, we deliberately had you generate an error. So, all right. So anyway, did anybody run into any questions or errors when you were playing around with the parameters for autoplot? Anything that you wanted to ask about? What does label size do? And change the plot color. Okay, first of all, label size changes the size of the labels, but only if you have labels enabled on your plot. So you have to set label equals true first, and then you can play around with the label size. So for example, I can set label equals true and label size equals 10, and I get gigantic, ugly label sizes, labels. Yeah, yeah, I have to have labels. So the color here is coming from so we tell autoplot, one of the arguments is data, and that argument tells the autoplot about the status of each data point. So we can just take a look really quickly at sample table and remind ourselves this is the table that assigns a condition to each of the samples. So we tell it color equals condition, and the condition here is used to color the data plots, the data points. These are the default colors for ggplot, which is being accessed by the autoplot function. If you want to change the colors in the plot, we have to go in and either change another argument for autoplot, or more likely add in another sort of what's it called, like a layer on top of this plot to change the colors. That's a little bit out of the scope of what we're doing right now, unfortunately. Doing visualizations using ggplot in R is like its own class. ggplot, there's a book on ggplot that's like a book. It's like 150 or 200 pages because it's so specific, and so you can make plots look like exactly what you want them to look like. But that means there are tons and tons of arguments and different layers that you can add. So I can send around documentation that we can read about ggplot, but if I were to try to go into that right now, it would probably take way too much time. What is theme classic? I believe that makes the background be white and sets the size of the plot. Amanda, did I say that wrong? Okay, yeah. Anything else? All right, so let's get into doing our actual differential gene expression analysis. So just to recap, what we found is that after we do normalization, we removed some of the signal coming from the technical separation, and we've improved our biological signal. So we're happy with our normalization, and we can go ahead and do our differential gene expression analysis. We've also run the walled tests already. Those are stored in our DE-seq dataset object, so we just need to access them and put them into some sort of human-readable format. So to do this, we want to create a DG output table, which is basically going to have all of the genes and whether or not they are differentially expressed, and the way that we know that is because we're going to report the p-value. We're also going to report the log fold change and the adjusted p-value, of course, very important. But we're going to just start out with our normalized counts table and add on to it with all these different columns. So the DG output table will have our expression data, and then it will have the different pairwise contrasts information from the wald test, and then we're going to add in some annotations, gene annotations at the end. So the first thing is we want to just start our DG output table, and all we're going to do is we're going to take our norm counts variable, which I'll remind us, it just holds a matrix or data frame of our normalized counts after having been normalized through a median of ratios. And we're going to just rename this to a variable that is useful to us as humans. So DG output table, and we're going to keep building onto this variable. So let's go ahead and run this. And I have here to take a look at the first 6 rows, which we just did anyway. But so this is the start of our DG output table. It has 12 columns currently because of the, these are just our samples, and then it has 6 genes and the normalized counts values and the ensemble IDs. So now we need to access those wald tests that we ran to get the pairwise comparison between all the groups. In our case, let's take a look at what the groups were by taking a look at the contrasts we set up initially. Remember, we set up these contrasts that are flight versus ground control and ground control versus flight. So we have 2 contrasts, which means that the wald test ran once on all the genes comparing flight versus ground control groups, and then once again on all the genes comparing ground control versus flight. So we want to grab the log 2-fold change, p-value, and adjusted p-value from the wald test, because the wald test reports all of those. Remember, we said wald test returns a p-value. It also returns an adjusted p-value because this is a bioinformatics program, and it helpfully reports log-fold change for us. This step takes a while, so we ran it in our Slurm script earlier. This is the code block that we use. We just iterate through all the contrasts using a for loop, and we say, pick out for the 2 contrasts, we want log 2-fold change, p-value, and adjusted p-value. And then we use that cbind function that we learned about to bind in those columns to our DG output table. Now, in the Slurm script, we did all of this and then wrote out that updated DG output table to a CSV file so that we can read it back in right here and take a look at it and continue updating it. Again, using the read CSV function. So let's go ahead and run this cell to read in our updated DG output table. Let's add a cell here to take a look at this DG output table that we've just read in. Let's do head just to look at the first few columns. So you can see we're starting out with the normalized counts, 12 columns, 1 for each sample, but then at the end, we've added in the log-fold change value for each gene for the flight versus ground control comparison, as well as the p-value and the adjusted p-value. And then we have the same 3 columns for the ground control versus flight comparison. So all in all, we've added in 6 columns the data that comes from the wald test. So now we want to add in some useful calculations that don't come from the wald test. We can just calculate these right on the counts data. We want to calculate the mean expression for each gene across the whole data set and the standard deviation across the whole data set, and then add those in to the DG output table. So the way we're going to do this is using 2 handy functions, row means and row SDS, which do exactly what you think. They calculate the mean of every single row and the standard deviation of every single row. On a data frame, note that row means takes the norm counts data frame as a data frame, whereas the row standard deviation requires us to convert it to a matrix using the as matrix function. We then are going to stitch these in at the end of our DG output table using the $ operator. So remember, we can use the $ operator to select a column out of select a column by name out of a data frame. We can also use it to assign a new column by just passing the name and then the $ and saying we want to assign this column name to this data frame. So let's go ahead and run this code block, and then let's view the first few lines of the DG output table and answer a couple of questions. So we can run head DG output table. So how many columns in total have we added in to on top of the raw counts matrix? So we started with, or rather the normalized counts matrix here. We started out with 12 columns, one for each sample. So far, how many columns have we added? 8 in total, that's right. And then you're right, Veronica, we just added in 2. The mean and standard deviation, and we had previously seen the 6. So what can you guys tell me, what is the average expression of the gene that ends in 31 in the flight group and in the ground control group by taking a look at this table? No guesses? Is everyone able to find the gene that ends in 31 here? So if we start here, the gene that ends in 31, scroll, you can say this is the 3rd row down, scroll all the way across. Oh, I think that question should be across all samples in both groups. No, this question is in the wrong place. We're going to calculate the group mean and the group standard deviation next. So let's come back to this question in just a second. Sorry about that. So right now we've calculated the total mean across all samples and standard deviation across all samples, which is somewhat useful, but we're interested in comparing the flight and ground control groups. So what would be most useful would be to actually know the group mean and standard deviation, so all flight and all ground control. And so the way we're going to do that is using some R functions that will break down our data set into just flight and just ground control samples, and then calculate mean and standard deviation on just those groups. And again, use cbind to stitch those columns into the end of the DG output table. So let's go ahead and run this cell. It takes a little bit to run because we are running a few different calculations here. First, we have to break up the data, and then we have to run mean and standard deviation. So we'll come back to this when this is done running, but let's start talking about the next interesting columns that we're going to add to our output table. So the next thing we want to do is add gene annotations. So we've been doing a lot of talking about the different types of gene ID databases and how they relate to each other. And the reality is that although our genes are listed as ensemble mouse gene IDs, and that's really useful because the ensemble database is so thorough, it's hard to look at an ensemble mouse gene ID and know anything about the biological relevance. And also, it's nice to have IDs from other databases, such as RefSeq, Entrez, and GoSlim, because there are other downstream analyses that sometimes require your data to be in a certain format. So to have, they say, oh, we only take RefSeq gene IDs or something like that. And then we're also going to add in pathway information. So as biologists, we know that genes don't act alone. So even though we might have a table where all the genes, we can treat all the genes as variables or as features, genes in the cell actually work in networks or pathways where they work in concert. They activate each other or deactivate each other. And so we can access certain databases which have lists of genes which are known to act in certain pathways. And these are also known as functional annotations because instead of looking at 100 genes, you can look at one pathway name and say, oh, you know, this set of genes is involved in mitosis or something like that. So that's all going to be very useful. Before we get to that, it looks like my command has finished. I hope everybody else's has as well. So if we again use head to check the first few lines of our DG output table, we should now be able to identify the average expression for the gene that ends in 31 in the flight group or in the ground control group. So that's just right here. You can see that this gene in particular has a mean count of 11 or almost 112,000 in the ground control group and about 76,000 in the flight group. All right. Did anybody not get this output? Not yet. Yeah, I was wondering about that. If perhaps having so many people running this at the same time could be bogging down the server. Is anyone else still waiting? All right. Okay, Aylin's just completed. Evelyn, just let us know when yours is done and we can move on to the next part. Because the one thing about Jupyter Notebooks is that you have to wait for the cell before to finish before you can run and see the output from the subsequent cell. Evelyn, are you saying okay that you'll let us know when it's done, or you're okay to move on? Oh, excellent. Okay. If anyone else is still waiting, please let me know in the chat. Otherwise, we're going to keep moving on. Okay, perfect. Did we already account for all the duplicated reads somehow or later? Amanda, were you going to say something?
Well, I was just going to ask for clarification from Felix. Are you talking about the gene IDs that map to the multiple gene IDs that map to the same gene symbol? Is that what you're referring to, Felix?
Actually, I'm thinking way back when we did QC, we found that there were a lot of duplicates.
We do not account for duplicated reads in our current standard pipeline. That is a pipeline that we are going to have to make, and we're only going to be able to do that if we have reads that contain UMIs. So in GeneLab, it's our standard to include UMIs in our sequencing, so that way we can account for duplicated reads. However, that is not, we accept data from any spaceflight omics experiment, and so not everyone will include UMIs. And we actually have not completed a standard pipeline to identify and remove UMIs. So that's a project that some of you guys are going to be working on over the summer. So we won't do that here, though.
So how was FastQC doing it? Because I remember at some point it said, this is the number of reads you have, and this is what you would have if you deduplicated.
So what it's doing there is, remember, that's only a sampling of the reads from that FastQC analysis. That's why I kind of take that with a grain of salt. So it's only looking at the first 200,000 reads, and it's only looking at the first 50 base pairs of those reads. So that number is really a drastic overestimation of how many duplicates there actually are. And what they do is they do a calculation where if they were to take just one set of reads from those duplicated ones that they identify as duplicates, if they were to just take one and remove all the others, that's what they're saying is their deduplication amount. So in doing that, though, they're not accounting, they don't have UMIs there, right? So they're not accounting for true biological duplicates. And also, if you were to deduplicate using the method that they're doing, because they're only looking at the first 50 base pairs of a read, you could have reads that are identical in the first 50 base pairs and then different in the next 100 base pairs, right? So we have 150 base pairs reads. And so in using the FastQC's algorithm of just looking at what they looked at, you would also could be throwing away reads that are indeed unique.
Got it. Thank you.
Thanks, Amanda. Okay, so the next thing that we need to do is for some of the databases that we're going to use, obviously there are different organisms, different model organisms, and also human data that people can use to access these databases. So we need to define an organism identifier, which will match the gene annotation database. So instead of having to type out mouse every time, we're going to assign the string mouse to a variable organism. This is also nice when you have a script that you're trying to automate. You can just have the user at the very top put in their organism and then use the variable organism in your downstream script. So let's go ahead and just run that cell, and then we're going to import an organism table which defines the gene annotation databases for each organism. So if we take a look at this, just by typing in organism table, we can see we've defined our organism as mouse. The reason it's in all caps is because it needs to match this organism table. And then this is the annotations database from R that holds all of the mouse or mus musculus annotations. So if we, I've just spoiled one of the questions here, but can you guys tell me what the annotation database is that's used for humans in this table? Yeah, exactly. So while the mouse database is called org.mm.eg.db, the human database, it just has an HS for Homo sapiens. Okay, so the next thing that we can do is to actually access this organism table from R to pull in all the different gene annotations for mouse. So we're going to start building an annotations table, but first we have to set up our annotation database, which we're calling and.dbi as a variable, and we're just using the BioC Manager package. Remember, we learned about this to install the annotation database. So again, this code is made to be automatable for any type of organism. So we could just run this line, BioC Manager : install the organism database that we're interested in. But because this code is from the GeneLab pipeline, it needs to be able to work for any type of model organism data that we get. So instead, we read in the organism table, and we tell it, grab from the organism table the organism type that the user input, which in our case is mouse. Call that a new variable, and.dbi, and then go ahead and install whatever was read in as and.dbi, which in our case is the name of this database, org.mm.edg.db, and just install that using BioC Manager, and then load it in using our library function. Okay, so let's go ahead and run this cell, and if anybody doesn't get this nice pink banner telling us that we've loaded in our org.mm database, please let me know. But now that we've loaded this database, we can use it to start creating our annotation database. So the way that we're going to add in all these additional gene annotations is by using the ensemble mouse gene IDs that we have in our data to find their matching IDs in the other databases. So the great thing about this annotation database that we just loaded is that it has mapping between all these different kinds of gene annotations. So it has ensemble genes, and then which ensemble gene matches which gene symbol, and then which ensemble gene matches which gene name, and so on and so forth. So we're using ensemble as the sort of the search term or the key type, and then we're searching through the annotation database that we read in, and we're searching for these 5 different gene types, and we're going to start building an annotation data frame called Annot. So here, what this row does is it starts an empty data frame, which just has the row names from the DG output table. And if we recall, the row names from the DG output table are simply the ensemble gene IDs. So we're starting an annotation table with ensemble gene IDs as row names, and we're going to fill in new columns here. For example, and again, we're using this $ operator. So for example, we're saying into this empty data frame, create a new column called symbol, and map the ensemble gene IDs to gene symbols and put them into the symbol column. So let's go ahead and run this cell. And we do get a little bit of a warning here telling us that in some cases there are multiple mappings. So an ensemble gene ID can have multiple matches in these different databases, and that's because of some of the limitations that Amanda has gone over with the gene annotations, where different people discover different genes at different times, and then also the different databases source from different studies. So this isn't perfect, but it still gives us a lot of great information and different gene IDs we can take to downstream applications.
And then Lauren, I just want to point out here, when it does map to multiple columns, if you go all the way to the right of your code block, you'll see that if there's multi-values, we take the first one.
Good point. Thank you, Amanda. So we're not getting a bunch of duplicates in our results.
Right.
Good point. All right, so let's just take a look at what we've done here. Using head, we can look at the first 6 rows of the gene annotation table. So you can see that we have our original ensemble IDs. And let's answer a couple of questions. So how many different types of gene annotations does our new table contain? Okay, I'm getting a few different numbers here. So the way that I would approach this question is just to look at how many different columns we've added in here and also to look at the column names. So um, we have the ensemble gene annotation. We then have symbol, which is the second one. We have the full gene name, which is a third. The RNA-seq, which is another gene annotation type, and then the entree ID, which is another gene ID type. So I would say 5 here. Can you guys tell me what is the gene name and symbol of the ensemble gene that ends in 28? Yeah, exactly. So the gene name is cell division cycle 45. The gene symbol is CDC45. Just to note, it is important. The gene symbol has a capital. The first C is capitalized, and that'll be important. That's how it was matched to the ensemble ID. So all of these gene IDs are case specific. What about the ensemble gene that ends in 31? Yeah, exactly. Beautiful. All right. Does it matter that that one is repetitive how it's labeled? No, it doesn't matter. This one just happens to have the gene symbol in the gene name. That's because many genes are first named like this with a long name, like cell division cycle 45, and then given a shorter name. H19 is a long non-coding RNA, so it doesn't, its functions are actually not fully characterized. And really what we know about it is that it's imprinted and has only maternal expression. So it's a transcript, it's not fully annotated, and so it was given the name H19. It didn't have a descriptive name like cell division cycle 45. I'm not sure what you're getting at there, Amanda. The H19 tell you about the, yeah, go ahead.
Disregard my second question. I was reading it wrong. I was thinking X chromosome silencing. That's not what it does. But I was just curious. Does anyone know what CDC45 does?
Well, I'm assuming it might have something to do with the cell division cycle. but were you looking for something more specific?
Yeah, exactly. So it's a cell cycle checkpoint. So it mediates how cells divide. So interesting that it's expressed in these animals. I'm curious to see if it's differentially expressed.
Yeah, good point. When you're studying disease states, you're always really interested in cell cycle genes because cell division is something that can go wrong in certain disease states. Okay, let's add in some more annotations, but these aren't new gene IDs. Instead, these are IDs which refer to protein-protein interactions. So we can use the string database, which remember all the way at the top of our notebook, we imported a database called stringdb. We also imported something called panther.db, which we'll use in a moment. So we're going to map our gene IDs onto string IDs based on, does that gene code for a protein that's found in this set of protein-protein interactions? So we're basically accessing the string database and then passing in the organism that, which is the mouse, obviously. Note here that when we access the string database and we want to map in the existing gene names that we have, we're basically searching the string database for protein-protein interactions that involve one or more of these genes. Note that string expects the gene symbol, not the ensemble ID. So that's one really good reason that we needed to map our ensemble IDs onto additional gene IDs. The string database can't deal with ensemble IDs, I'm assuming. And so we're passing in, instead we're searching using the symbol gene IDs. So we're going to go ahead and search the string database, and we're going to use a new function called dplyr. So this is a library which is designed for merging together data frames. So this is a little bit more flexible than cbind. So we're going to map in together our original annot table with our string data frame. So let's go ahead and run this cell. Did we just download these annotations from the web, or were they installed in HPC? So I believe we're accessing the web right now. Or, I mean, what I mean is that I believe we just downloaded the annotations table for mouse when we ran this cell. Go ahead.
Yeah, for sure we did. And you did that when you ran the BIOC Manager install. So that's when you actually installed that from the web, and then you loaded the library. And the reason that we install it every time we run this script is because annotations are kind of updated all the time. So we want to make sure we're getting as much annotation information as is available.
Right. So everything else we didn't install, like you said, they were installed on the HPC, and we just loaded those libraries. But Stephen installed everything back at the beginning of this year, which means that there was plenty of time for a database to change. So in this case, we just installed it. And then when we installed it, now we're searching a sort of locally held database. So we're not like searching the web, we're searching a database that we just installed. Okay, so has everyone's string mapping finished running? Is anyone still waiting for this cell to run? Great. Let's take a look at the first 6 rows of our string annotations table. So this is the table that we mapped onto, or we stitched onto our annotations table, but let's just look really quickly at what a string annotations table looks like. So here, like I said, we searched the string database using the symbol column, and we came up with a bunch of what's called GhostLim IDs. No, rather, I'm sorry, that's what we're going to get from the Panther database. We came up with a bunch of string IDs, each of which refers to a different protein-protein interaction that must include the gene here on the left. So just to illustrate what I mean more concretely, the warning, oh, no, I didn't mention it already. Thanks, Felix. So 28% of our identifiers, in this case, the symbol gene IDs, weren't found in the string database, which is fine. That just means that there isn't an annotated protein-protein interaction set in string for 28% of our genes, which could be maybe those genes are not well annotated, so we don't know which other proteins they interact with, or they just haven't been put into the string database yet. Sounds like a lot. Yeah, it is. Keep in mind that a lot of these are non-coding RNA, for which so we're talking right now about physical interactions between proteins, which are derived from, pull down assays and things like that. Because non-coding RNA are not, obviously, they're not coding, so they're not translated into proteins. So those automatically are not ever going to have a protein-protein interaction. In general, non-coding RNA control transcription or affect genes at the level of RNA, not at the level of protein. So that's one reason for a lot of genes not being in string. And then there are just, because the way that we get physical protein-protein interaction data is that people in labs have to do those pull-down assays. We haven't mapped out all the possible protein-protein interactions in the cell. So it's definitely limited. All right, so the last set of gene identifiers we're going to add in are the GhostLim IDs, which we're going to import from the Panther annotation database. And oh, that's a good question, Amanda. So a pull down assay, there are different. So I've never done a pull down assay, so let me just stumble through this here. No, I know what it is from reading about it in papers. If you are interested in a particular, so there are different ways to do this, but the basic concept is that you can isolate from a cell, you can isolate some protein and figure out what other proteins are bound to it at that time through a variety of biochemical experiments. And in that way, you can say, okay, the protein that I'm interested in interacts with all of these other proteins. And you can do that depending on which assay you're using, it's more or less scalable. So it's simply a way of taking biological material, isolating proteins, and saying which of these proteins are bound to each other. That's because proteins, the assumption here is proteins tend to affect each other through physical binding. So you can say if these proteins are bound together, that means they have some kind of interaction, they're affecting each other in some way. Activating each other or deactivating each other or something like that.
Oh. If I use the whiteboard?
Yeah.
So just as an example here, we have a protein X and we have a protein Y. And let's say we know what protein Y is, but we don't know what its binding partners are. So we don't know what it interacts with inside of a cell. Pretend like we're in a cell here. And so since we know what protein Y is, we can create an antibody that's going to target or latch on to protein Y. And of course, we have to make sure that it's unique to protein Y, so we don't want that latching on to anything else, or else we'll get off-target effects. And what we learned from the library prep when we used this antibody technique to pull down ribosomal RNA, we can also, in addition to this antibody being specific to protein Y, we can also biotinylate this antibody so that way it's going to be magnetic. And so then we introduce this antibody that has this biotinylation to it into our cell system, right? Of course, we have to bust open our cells to do this. And then we add in this antibody, and that when it bumps into Y, it's going to latch on. And then we use the magnet to pull it down. And then we can do a protein assay, like look at gas chromatograph mass spectrometry, liquid chromatography. There's a variety, do a Western blot analysis. There's a variety of different ways to assess the proteins that are in here. And then we could see all the binding partners of protein Y. So then we can identify like protein X is going to be one of its binding partners in that way. And then once this interaction is known, that information will be published by the researchers that discover the binding partners of Y, and that information will go into that string database, and then we'll be able to pull it out.
Right. No, thank you, Amanda. And I just want to address what Felix said. It is a lot of work. Generally, we're getting this information from people in labs who are interested in a specific protein. So they did one of these assays and they published those data, and then that went into a string database. So it's kind of a one-off, we're getting these data from multiple different places. So that's why 28% of our genes aren't found in the string database, because someone just hasn't done that work yet, or the non-coding RNA situation. For you to say something else.
All the wet lab workers for for getting us that information. Wet lab bench work is not nearly appreciated enough, hence why I switched to dry lab.
Absolutely. Can we do that with a computer? That's a lot of work. I think we can make inferences, and we can generate hypotheses with a computer that we can test in the lab. I don't know of a computational program that, I'm sure there are computational programs that infer protein-protein interactions. In fact, I've seen databases, there's a database called MERNet, which has microRNAs and long non-coding RNAs, and it has inferred relationships, and it'll give you a percent confidence for the relationship. And this is all based on like literature mining. So generally the thing is I don't use those when I'm accessing that database, because I don't want to have to defend that to a reviewer. Like, some of my data is actually inferred and I don't actually know, we have the confidence level, but it wasn't validated in a lab. So that's always the problem, Felix, with trying to convince biologist reviewers on a paper that something that you did in a computer is the same as doing it in a lab.
Yeah, but you are right. There are tons of databases and programs that if you know the molecular composition of your protein, so if you know the amino acids that compose your protein, right, we know what functional groups are on those amino acids. And so based on what we know about how those functional groups interact with other functional groups, we can make predictions about which proteins they could possibly be interacting with and make models like that. We can also, this is used a lot, that type of modeling is used a lot in drug discovery. So for instance, when I was in graduate school, I was working with protein kinase C, the atypical versions of these, and they have a binding partner called PAR6 that they need to interact with in order to promote tumor growth. And so we were looking for a drug that could stop that interaction. And so what we did is we knew the amino acid sequence and what protein kinase C and PAR6 looked like. So then we just submitted that into a database that looked at all already FDA approved drugs to see if anyone would likely prevent that interaction from happening, and was able to find some candidate drugs that ultimately we were able to put on into clinical trials for the cancer that we were looking at. So that happens all the time. There's also other things for like RNA and how RNA folds and how it opens up. So that way we can also do pull downs with RNA like we did with ribosomal RNA. And like one program I know is called Foldit. It's an open source program. It kind of makes it like a game. And it's also doing a machine learning thing in the background to better predict how the RNA, based on the different sequence that it has, and again, if there's any modifications to that RNA too, how it folds and what areas are accessible. And so that way we can get better and better predictions using these machine learning algorithms types of techniques.
That's great insight and experience. Thank you, Amanda. Yeah, absolutely, Felix. So let's go ahead and add in the GoSlim IDs, which are every GoSlim ID stands for gene ontology. I don't know what the SLIM is for, but this is now, instead of protein-protein interactions, we're now looking at sets of genes that are known to interact together. So unlike the string IDs, these gene sets could include non-coding RNA that are known to, for example, modulate the expression of some coding RNA. So we're accessing the Panther database with the organism that we set up to be mouse. Here, interestingly, the Panther database only takes the entree ID. So we had to search using symbol in the string database. Now we're searching with the entree ID in the Panther database. And we're going to add in a column called GoSlim IDs to our Annot table, again using the $ operator. So let's go ahead and run this cell, which will access the Panther database for us. Well, the Panther database has already been downloaded, so it's just going to search through it for us. I guess this is going to take a couple of minutes to run as well. The organism name we use is based on the Panther name used. Yeah. So Panther has specific naming conventions, and we're just aligning with them. How would you check for the non-coding RNAs, Lauren? I don't know exactly what you mean by check, but I can start to answer that question by saying that there are lists of known non-coding RNA. Yes, so I would go and say, so I have my string database. I would take all of these gene names and then figure out which gene names from the original table aren't found in string. And then I would compare the genes that weren't found in string against some known GTF file of non-coding RNA. So there are long non-coding RNA GTF files or microRNA GTF files, and so I have a combined file that has like all the most recent non-coding RNA names, and I'd say, okay, how many of my genes that weren't found in string are actually non-coding? Does that answer your question? All right, so on my end, the Panther search finished. Is anybody else still waiting for it to finish? All right, excellent. So let's take a look now. We're just going to look at the first row of our new gene annotations table, because the reason we're only looking at the first row is because the GoSlim IDs take up a lot of space. So we're just going to look at the very first gene here, which ends up for example, not being found in the string database, even though it's a protein coding gene. But it is found in a whole bunch of different gene ontology gene sets. So each one of these IDs refers to a list of genes that are known to act in concert with each other to effect some sort of biological change. So these genes are involved in mitosis, or these genes are involved in actin cytoskeletal assembly, or so on and so forth. So there are, let's go ahead and answer these questions before I spoil them. What is the name of the gene that we're looking at here? We're just looking at one gene. What's the name of it? Yeah, exactly. Very good. It's a G protein. How many biological functions or gene sets involve this gene or this gene? Amanda, do you see Nicholas's question? Oh, did everyone's?
I'm actually having the same issue when trying to sort it out too.
Oh,
It's the only one since no one said anything.
oh, you guys are all so polite. Okay, my kernel didn't die. I don't have it. Okay.
It's because just Lauren's was running and she made all of ours die. All the resources.
Okay. Okay, so what we're going to do is everyone whose kernel did not restart, like me and Felix, don't do anything. Everybody else, please scroll back up to the place, remember where I said we created an error? I want you to comment this cell out, the one where we reran DSeq dataset from matrix. Sorry, is someone talking? Were you going to say something, Jonathan? No, okay. Oh, okay. Please comment out this line so that we don't run into an error. This is the one under the challenge where it says, see what happens if you run DSeq dataset from matrix without first converting the data to integers. I want you to please comment out this row with a hashtag. This is and then please come back down to where we were. Where did I find it? I'll let you know. Here we go. And then you're going to select this cell, make sure it's selected, click on the number by it, make sure that you have the two blue bars to the left, and go up to kernel, and then restart kernel and run up to selected cell. Is anybody having any issues doing this? Yeah, it probably will take a while, Felix. I'm staying busy, so I think it's working. Okay, you can also scroll up and kind of watch it run. So I guess mine isn't going to do anything, but you can scroll up and if it has a little asterisk by the cell, that means it'll run. It's running. So I ran into an issue. It's failing to open files.
Yeah, I'm getting that same issue.
Okay, where is it? Like,
Mine starts up with a sample table. Is that where your guys is running into that issue too, where it's trying to read in the metadata CSV? Yeah.
okay.
It says it's a stale file handle.
I'm gonna see if I can replicate this. Dare I? Okay, I've restarted the kernel. Oh my goodness. Okay. I wonder if something has happened on the server end. Is Steven on?
I don't think he is. I've been poking around on the server and things are moving really slowly.
Okay.
So I think we can blame the server if that's for safe.
Okay. I think what we should do is we have a break in 15 minutes. So for the next 15 minutes, I'm going to switch back to the lecture and teach us how to do visualizations. And then if everything is still not back over the break, then we can talk about what to do. One option is that I could run everything locally on my end and just show everybody what's going on. That's not as fun as you guys running it yourselves, but we are almost to the end. So, in the meantime, let's get back to the lecture. And this is the most fun part of the lecture anyway. Steven says there are some power users hogging resources. Good to know, I suppose. Steven, would that explain this issue that we're getting where it says we can't even the stale file handle thing?
So I looked it up and it looks like it's an issue with the mount point. So all of the data that we're pulling from the GPFS, whatever that acronym is, storage space on the cluster. Yeah, and I just did a DF and it looks like something happened with that mount. So the mount to the storage space where the data is fell off.
Oh no.
Usually you can remount it fairly easily. Stephen, do you think you'll be able to do that?
Looking into it. All right. We appreciate it. Thank you, Stephen.
Yeah, thank you, Stephen.
All right. So let's just get back into the lecture here. So as we know, when we're running differential expression analysis, we start with thousands and thousands of genes. So it's easy. It could easily happen that we're going to have hundreds or thousands of significantly differentially expressed genes. And that is a little bit difficult to look at using the human eye. So it's really nice if we can interpret some sort of visualization or figure. So two common visualizations for RNA sequencing that we're going to use are the heat map and the volcano plot. So a heat map, here's an example of a heat map. So you start with a numerical matrix where in our case, we're going to have samples across the top as column names, and then we're going to have genes along the side as row names. And then within the matrix, you have your numbers, which in our case are the normalized counts. The goal is then to map the values in the matrix onto some kind of continuous color chart. So in this case, we're mapping these values onto a color chart that goes from blue or purple all the way up to red. And the values are being mapped into the little intervals along this continuous axis, which makes it nice to look at and see relationships between samples and between genes, which we're going to get into in a minute. In this particular heat map, the rows have been clustered based on the similarity of the values across all the rows. So you'll see instead of in our original matrix, which went gene 1 through gene 18, here in the clustered matrix or clustered heat map, the genes have been rearranged based on similarity. So you can see, for example, these genes, which weren't near each other in the original matrix, all have fairly high expression in this group of samples, which brings me to pointing out that the samples or the columns have also been clustered based on similarity of values. So you can see patterns starting to emerge based on how similar samples are to each other in terms of the values of gene expression in different genes. So for example, we have 3 main clusters of samples. If you look at this little clustering dendrogram up here, the rightmost branch has high expression of the first group of genes and then lower expression of the last group of genes. And there are 2 main groups of genes you can see here too. And then the middle group of samples has lower expression of the first group of genes, and then it gets kind of higher in the middle, then lower with the last few genes. We call this the legend or the color bar, and these colors have been mapped to the range of values in the matrix. Now, you may have noticed that the color bar here goes from -2 to 2. Now, this might seem a little bit artificial. Do you think that these data values or any real data actually naturally fall between -2 and 2? Probably not, right? So we know, at least in our data, the range of values is gigantic. But think about, if we took our data, which we saw can range from 2 all the way up to almost 200,000, imagine trying to map that many different values onto a color bar. I'll tell you what'll happen. It will crash your server. It will crash your Jupyter notebook. So instead, it also is very difficult to even look at a plot like that, because the higher values will dominate the plot. So and the lower, actually rather the lower values will dominate the plot, because most genes have close to 0 expression, right? So your entire plot is going to be blue with a tiny little bit of red. Very difficult to see actual relationships between genes in that way. So these data have been scaled. If you try to create a heat map, this particular heat map, without scaling your data. So scaling here means taking these values which fall far, far away from each other in value, and then mapping them onto a much smaller area of, in this case, of the plot. This is just kind of a visualization of what happens when you scale values. You can see that after scaling, the values maintain the same relationship to each other. The yellow value is up here a little bit, and the green and blue values have the same relationship, but they are mapped into the dispersion is much, much less. They've been mapped into a smaller area of the value plot. So we are maintaining the proportion of the differences between the values, but not the actual literal values that we started out with. Pre-scaling gene count values can actually range from much higher than 30,000 to 1. And these larger values can just skew the data non-representatively. Larger values also in techniques like PCA, values with really large, sorry, I've forgotten the word for it. If you have a gene, for example, that spans a very large range, that's the word I'm looking for, then that's going to eat up a lot of the, PCA is going to think, oh, all my variance is right here in this gene. But after scaling, you've maintained the proportion. You've maintained the relationship between the values, but all of the values have been brought into similar proportions so that they can be reasonably compared, and so that your computational tools are able to make use of them. Stephen says it should be working now. Great. Thank you, Stephen. Why don't we go back, and in the last 5 minutes, let's see if we can make sure that our hub is working. So everybody go up and do restart kernel. And then let's run the first few cells and see if we can import these files. All right, I was able to import the sample table. Is anybody else running into issues? Thank you, Stephen. That was amazingly fast. We really appreciate you being here.
Hey, Lauren, do you want to have everyone do what you had before and just start everything running before
Yes.
the block you're on? And then as it's running, maybe you can get back to your stats lecture in the last 3 minutes before the break.
Yes. That is a great idea. So everybody please scroll down to the cell where we ran head and let's see, I'll put this text in the chat. Select this cell and then run restart kernel and run up to selected cell.
And then just a reminder for those of you guys who didn't put those hashtags next to those DDS tests that we did, make sure you do that before you run, or else it'll stop there because of an error.
Yes, thank you, Amanda. Good point. Let me know if you are running into issues. So after you've run restart kernel and run up to selected cell, you can scroll up and make sure that all of your cells have asterisks by them. If they end up not having asterisks by them, that means you've run into an error. So see, mine has I can see a cell where it says head DG output table and it has a number by it, which means it's run. The next one, which is one of the ones that took a long time to run, has an asterisk by it, which means that it's still running. Is anybody having any issues with this? Okay. Okay, great. So I'll just, in the last 2 minutes, get started on volcano plots and we can finish up after the break. All right, so after you run a differential expression analysis, we've seen we get our genes, we have our log fold change values, we have p-values, and we have our adjusted p-values. And we can take these values and turn them into a volcano plot. And this is kind of a lot to look at, especially if you haven't seen a volcano plot before. So I'm going to break this down for you. First of all, the axes on this plot, the x-axis is the log fold change values. The y-axis are the adjusted p-values, but we take the negative log of the adjusted p-value. The reason that we do that is because adjusted p-value or p-value can be arbitrarily small, and trying to visualize something that can be, 0.00000 whatever is a little bit difficult. So instead, when we take the negative log, we now say, instead of a low value being good, which is what we say about a p-value, now a high value is good. So after taking the negative log, the higher the negative log adjusted p-value, the better. So we plot negative log to log fold change. Does it add a small number to all the adjusted p-values? That is a good question. I imagine that it has, so that's called a pseudo count. I imagine that it has to do something to take into account, for example, the 0 value here. So I imagine it does add some kind of a pseudo count, but that wasn't upfront in the documentation, so I don't know for sure. I do see that it is 2:30, so let's go ahead and take our break, and everybody can be back here at 2:50. Thank you, everyone.